I'm here today to take you through the process of PCR. You've probably heard of PCR before with COVID-19 tests, but have you ever wondered what it actually is or does? PCR stands for polymerase chain reaction, and it's a tool used to make many copies of a gene through repeated heating and cooling. PCR basically allows for the cloning of DNA strands, which in turn helps us with the identification and detection of gene sequences using visual techniques based on charge and size. Its ability to make billions of copies of a DNA strand gives it an array of applications, including research and genetic studies, forensic analysis, and medical diagnostic. If you watched a DNA replication video, the PCR process probably sounds quite familiar to you. The first step would be identifying the DNA sequence or gene you want to replicate and place primers, short fragments of single-stranded DNA that match the start and end of the target gene in the DNA pool. The primers mark the starting point of DNA synthesis. The second step would be to heat the DNA pool in order to make the double helix DNA into single strands, basically separating the two strands apart. Once they're in single strands, the temperature is lowered and the primers inserted previously can bind, also known as anneal, to their complementary places, which will mark where transcription should start. Now, the enzyme Taq polymerase binds to the primer that was inserted earlier and begins transcription. This is done by the Taq polymerase adding complementary nucleotides to the strand, like so. You now have two pairs of the section of DNA you want to be cloned. This cycle is repeated through the heating and cooling of temperatures to create numerous copies of the desired DNA over a short period of time. An advantage of PCR is that one copy of the target DNA sequence can turn into millions of copies in a short amount of time.